All right, thanks, Sylvia. Today it's a pleasure to welcome Martyn Kelly to our Diatom Web Academy. Martyn tells me he's been looking in microscopes since his early teens. And after getting his PhD at Durham with Brian Whitten, he spent a couple of years lecturing in Nigeria, of all places. But where he comes from us today is the middle of the UK, where he represents Boburn Consultancy, which he tells me is celebrating its 25th anniversary during this COVID pandemic year. Otherwise, there'd be big parties going on, I bet. Martyn also serves as a member of the Council for the International Society Diatom Research, but his work has really been heralded in developing multiple approaches and indices for assessing aquatic health, especially with diatoms. One of the things that he's been really involved in is integrating approaches between the UK and the EU, using this new regulatory landscape we're in to develop and support the use of nutrient criteria across Europe. One thing I do want to point out is Martyn runs a really nice blog called Microscope and Monsters, and I'm going to just pop that right there into the chat box if people want to check that out. It features a lot of his artwork as well as some very nice writing directed at sort of a more lay audience. So Martyn, with that, I'm going to turn it over to you, and thanks again for doing our presentation today.
Okay, thanks a lot, Mark. So I've just got to share the screen again. Okay, so that's everyone can see that, can they? Yes.
Yep, looks great.
Good. Okay, so talk is based around no, not sharing. That's it. Talk's based around this paper, which a group of us produced over the summer. It's a group of us with experience of ecological assessment on both sides of the Atlantic. And we held a series of Zoom meetings over the summer discussing how periphyton was used in the EU and the US. And this paper is the outcome of those discussions. And this is a graphical abstract to summarize the paper. The core message is that emerged from our discussions was one of ensuring consistency at different levels within federal structures that spanned huge biogeographical variations. So this talk is going to be in 4 parts. And one of the things we realized quite early on during our discussions was many of the differences we talked about were driven by the wording of the legislation that we were dealing with and by the structures within which it was enacted, rather than necessarily by scientific differences. So what I'm largely going to do, I guess, is sort of focus on the European side of this talk, sort of try and explain how things are done in Europe, and as far as I understand it, how they differ from what happens in the US. And if I'm going to do that, I need to start off by telling you a little bit about the Water Framework Directive, which I'm sure many of you have come across in papers and talks from people in Europe. So I'm just going to summarize it very quickly on a couple of slides. So the water, oh, all of my colors have gone weird. Never mind, I have to push on. Water Framework Directive, the core concept of the Water Framework Directive is ecological status. It's important to realize that ecological status is a condition of a water body. It's not, it's a sort of a condition of individual components of the biota, but really at a legislative level, it's a property of the water body itself. So we have to consider several groups of organisms when we're assessing ecological status. And we assess ecological status on as 5 classes: high, good, moderate, poor, and bad. And these are roughly equivalent to the biological condition gradient. We talk a little bit more about that in the paper. And the worst of the individual assessments defines the final status of the water body. And this is a controversial one out, all out rule. So if we assess fish and invertebrates and algae, and the fish and invertebrates are high or good status, but the algae are moderate status, then the status of the whole water body would be moderate. And if the biota comes out as high or good status, which means no or only a slight change from what is expected. That's fine. The water body is deemed as sort of appropriate for sort of long-term sustainable water resource. And if it's less than good status, then the member state is expected to apply a program of measures to bring the biota back to good status. And the Water Framework Directive sort of fits into a broader sort of level playing field for competitiveness across Europe. And what it would mean is good status in an Irish lake should equate to good status in coastal waters off of Cyprus. And that in turn should mean that fishermen in Ireland and Cyprus are expected to be conforming to the same standards or same environmental standards in their work. Sort of a key aim of the European Union. So I said we had to assess different components of the aquatic ecosystem. These are called biological quality elements. Invertebrates specified. The phytobenthos is what the periphyton is referred to in Europe, and it's bundled together with the macrophytes as a single biological quality element. Then for phytoplankton and fish. So everybody should be monitoring all of these or given a good reason why not. So generally be accepted that in a low order, that in a sort of low order upland stream, phytoplankton assessment isn't going to be that useful, so you don't need to do it. But you've got to assess those or give a good reason why you're not assessing them. And macrophytes and phytobenthos, I should add, are usually sort of, is treated as a single quality element, but they're usually assessed separately. And member states are also expected to assess hydromorphology, physicochemical supporting elements, that would be nutrients, pH, oxygen conditions. And the priority substances and priority hazardous substances refers to toxins, pesticides and heavy metals, so on and so forth. And all of this takes place under a political framework called subsidiarity. So matters ought to be handled by the smallest, lowest or least centralized competent authority. It's a general principle, came in with the Maastricht Treaty in 1992. So the EU only acts where action of individual countries is insufficient. And as many of the water bodies are straddling national boundaries, in some cases they are national boundaries, there's a good case for the EU to step in to ensure equivalent standards are applied throughout a catchment. So the way subsidiarity is working is that the member state sort of sets up the monitoring framework, does the assessment of ecological status, designs programs of measures, enforces sort of water management steps, and reports progress to the European Commission. The European Commission, on the other hand, ensures consistent application of the Water Framework Directive and ensures compliance. And there's something called the Common Implementation Strategy, which brought experts and the European Commission together to work out how to decode this large piece of legislation and apply it. And the working group responsible for ecology is EcoStat, and Sandra Poikane, who's one of our co-authors, and myself have been involved with EcoStat for a number of years. So this second point I want to talk about, something where the EU and Europe and EU and the US differ quite a lot, is in the choice of metrics that we use. And generally this emerged from our discussions over the summer. In Europe, well, first of all, it's difficult to make absolute categorical statements about what happens in the EU and US because there's exceptions on both sides of the Atlantic. But generally, the EU countries were preferring to use single metrics based on composition alone or relatively simple multi-metrics. And also most of the metrics that we're using were developed before the Water Framework Directive. The IPS is particularly popular, used in more than half the countries. It's two metrics developed by Eugen Roth's group in the early 90s, quite widely used. They're often bundled together as a simple multi-metric. The TDI, which I developed in the UK, is used in 5 countries, and 11 other indices are used in 1 or 2 countries. One of the striking differences between EU and US is relatively few are measuring abundance as part of their phytobenthos. Well, actually only one country includes abundance in their phytobenthos method. And some other countries are including cover assessment as a sort of a proxy for the quantity of algae in their macrophyte assessments. I'll talk a little bit more about that in a moment, but I think what happened is the reason why we've repurposed existing water quality indices, whereas the US has tended to develop metrics from scratch, is we had very tight deadlines. So 6 years between the Water Framework Directive becoming law and monitoring and assessment being expected to start. And also the one out or all out rule in effect creates a huge meta multimetric spanning all trophic levels and also physical and chemical measurements. So phytobenthos doesn't have to capture the whole story. And also, I think a lot of the focus, especially in the early years, was on setting up shared benchmarks. So I think that's pretty much why we felt, you know, going back to those early years, there was a sort of a feeling that we had quite a lot on our plate. And if we could pick up a existing metric and sort of batter it into shape, then that would do for now. So this issue of setting benchmarks took up a lot of our time in the early years. And so it's something that I'm going to focus on for the next few slides. I'm going to break it down, first of all, into sort of the reference benchmark and then into regulatory boundaries. So a word that's cropped up a couple of times already and will crop up again as the talk goes on is this word comparability. The EU wanted consistent application of the Water Framework Directive across the entire territory. And the way this was going to be achieved, or the way this is achieved, is by expressing all ecological status assessment results on the same scale, which is an observed versus expected scale, the ratio, the ecological quality ratio. And the idea was that the expected value, the denominator, would always be the same. It would be the reference condition, the no impact site. And so the directive specifies that we should be setting up sort of types, different water body types, recognizing that sort of upland rivers and lowland rivers, you would have different expectations, hard waters and soft waters, you would have different expectations. And then the expected value would refer to the sort of the no effect condition within each of those types. And no effect defined as no or very minor anthropogenic alterations. And an awful lot of time has been spent sweating over just what very minor means, as you might imagine. How we set common benchmarks is originally set out in the grey literature and lent heavily on this paper that you're probably familiar with by Stoddart et al. In fact, Richard Johnson, one of the co-authors of that paper, working in Sweden, was one responsible for sort of setting us on course for reference conditions in Europe. The aim is for a consistent approach, not just between countries, but also between water body types. So whereas historical condition might appear to an audience of diatomists to be a fairly obvious choice for reference conditions in lakes, it's not something that can be easily applied in other water body types, rivers and coastal waters. And so the focus has been on trying to find the least disturbed condition, which we can sort of equate to the no observable effect concentration from toxicology. The option of best available condition was discussed, but in the early years it was not recommended because of this concern, what happens if your best is not very good? And bear in mind, we've got some very densely populated parts of Europe, such as the Netherlands. So if we felt that if we went for best available condition, it might be some distance from a true sort of no effect concentration. And then finally, the Water Framework Directive always falls back on this term expert judgment. And expert judgment can mean experts making a judgment based on all their experiences, but sometimes it means guesswork, and sometimes it means defaulting to a number that the bureaucrats aren't going to argue with. So on the whole, the Water Framework Directive prefers to go with the least disturbed condition if possible. And over time, something called the European reference approach developed, and a sort of mixture of sort of objective and subjective processes. It came up with some criteria for land use and chemical thresholds that sort of roughly equated with the no effect concentration. So this is an example of its application to lakes, no point sources of pollution, low population density, low proportion of agriculture. You might well see with a threshold of 20% agriculture what we mean by those discussions about what very minor might mean, and sort of no introduction of fish or commercial fishing. Similar principles adopted for other situations, sometimes, as I said, supplemented by chemical thresholds. Now, I'd said that we were expected to set up types for determining reference conditions. The UK deviated from this approach of type-specific reference conditions for many of its biological quality elements, not just the periphyton. And instead, we tried to use equations to predict expected values from key non-pressure variables. And I'll show you how we did this for the periphyton. So the map on the left is the data set we use to set up the diatom tool. Sample sites sort of as far as possible scattered across the UK, that's Great Britain. We also had sites in Ireland too, but they're not shown on this map. And the map on the right shows reference sites in relation to the whole data set. You can see there's a big gap in the middle of the country where there simply weren't any reference sites. And these were mainly lowland calcareous catchments, sometimes with overlain with drift geology. And particularly important to note, these have often been used for agriculture for over 4,000 years. So the whole sort of idea that we could go out and find reference sites in lowland UK and indeed in lowland Europe was maybe in retrospect a little bit fanciful. So anyway, I'm jumping ahead of myself a bit. What we did with our initial reference data set was to model the TDI in the reference subset against key non-pressure variables. And in the case of diatoms, the only non-pressure variables that we had that were explained a reasonable or significant proportion of variation was alkalinity and season. So we set up an equation which incorporated alkalinity and season that enabled us to predict a reference value for each sample. But look how the data on this graph starts to tail off at about 100 milligrams per litre calcium carbonate. Now that's roughly the point where limestone starts to make a significant contribution to catchment geology. So we are effectively steaming ahead with a legally enforceable regulatory program on the basis of an extrapolation. So that was far from ideal. We probably made some fairly bad decisions in the early years, and we've sweated over finding the better alternative subsequently. And given that I've just been quite rude about the benefits of best available, I have to confess that we came back to the idea of best available because, quite frankly, that was all we've got. So this paper, published in Ecological Indicators, part of the same special issue as the review paper, sort of explains how we've sort of repurposed the best available approach with 20 years experience behind us. So this is a similar graph to last time insofar as we're plotting the TDI against alkalinity. But the difference is this time we're not just looking at the subset of reference sites, we're looking at the entire data set. Now on this graph, we've got sort of 3 pairs of lines. The blue line is the original reference data set, which I've already introduced you to. The black line is a sort of an interim reference model that is probably, sooner we forget about it, the better. And the red line is the new reference model that we developed. We developed that, I mean, this was a group of us now working. Steve Juggins has played a key role in statistics over the years. We're now working with Jeff Phillips and Nigel Wilby in the UK as well. So once again, we tried different non-pressure predictors. Alkalinity came out as the only one, apart from season, that had a significant effect. But then, you know, I've already mentioned that there's a human factor here, because limestone and agriculture are interwoven across the European landscape. So we need to recognize this human factor in the alkalinity gradient, sort of the role of agriculture. So we did this by including nitrate in our predictive equation. Did I mention that, perhaps I'm not sure if I did mention, should have done, this time we're using quantile regression rather than straightforward linear regression in order to fit our equation. So we're sort of trying to model a lower quantile, the lower quantile being some sort of balance between the lowest values we've got whilst retaining some statistical robustness. And we've included nitrate in the equation, but when we're predicting reference, we would set nitrate to a background concentration, so typically 0.1 milligram per litre. And once again, we've got a small effect of season, which doesn't make a huge difference to the equation, but it's sort of an easy variable to include, so it's still there. So to reflect on this sort of setting the reference benchmark from the European perspective, we realized early on the denominator in the EQR needs to be robust. So it's easy to sort of set yourself a very sort of pure reference concept, but actually if you're going to use it as the basis for a regulatory system, then it needs to be robust, which means you need several reference sites per stream type, per ecoregion, and because this is a responsibility that's devolved to individual countries, per country as well. Following on from that, the least disturbed condition isn't feasible across much of the European landscape. So what I've told you about the situation in the UK applies to a lot of sort of Central Europe. Sometimes some countries are in a much worse position than the UK here. But also, I think the UK has demonstrated for several organism groups, not just the periphyton, that site-specific predictions are a viable alternative to type-specific reference values. And in fact, you could say that because we're including dates in our equations, these are actually sample-specific predictions. It's a paper I published with some others, Steve Juggins and some colleagues in Romania a couple of years ago, that said that when you're using predictor variables such as alkalinity and in this case conductivity, you really need to date stamp them due to the effects of climate change. So we are always using conductivity as a predictor variable, but using the 2010 data to predict the reference values, simply because the conductivity in many Southeast European lakes is creeping upwards at a sort of a rate you can sort of easily notice. So best available condition, I still think you should be using it with caution, especially with small data sets. But the state we're in now, after 15, 20 years of monitoring, is that many EU states have got quite large data sets, so it is worth revisiting. And what I didn't say above is that if you use best available, then you need to accompany it with a value judgment to decide whether you really are predicting reference or whether you're actually better off predicting a lower boundary state. So in the UK situation, we were fairly confident that the values we were predicting could be used as the denominator in the EQR calculation. But in the Romanian example, we felt that our quantile regression was leading us to the boundary between high status and good status rather than sort of the true sort of reference value. So having defined the denominator in the EQR calculation, what about the other status class boundaries, and in particular, good moderate status class boundary, which is a key boundary for regulatory purposes? Because if you're below the good moderate boundary, then you're expected to put a program of measures in place, which can be sort of expensive. So the early Water Framework Directive grey literature set out some options. The first of these was, look at your sort of ecological gradient and look for step changes amongst it. We had some vigorous discussions over the summer. Jan Stevenson's a big fan of step changes along a gradient, and it does make intuitive sense. If you've got a step change, then you probably should be using it. Trouble is, when we define types very broadly, if you often find that the step changes, we often find, well, it's difficult to find step changes that are sort of robust enough that mean that you're not going to be plagued by false positives and false negatives. And also, I think because we've set up many of our metrics using linear modeling, many of the discontinuities in the data sets that would lead to these step changes are sort of being naturally ironed out. When you've got metrics based on 25 or 30 species, a step change in one species is sort of compensated by sort of changes in 2 or 3 others, and you end up with sort of a linear relationship. So a second option that was suggested was that we could use the point where 2 complementary metrics cross over. Now, of course, many cases in Europe we're not using multi-metrics, but we can decompose metrics like the TDI or the IPS to yield sensitive and tolerant taxa. And then pretty much as a last resort, this early document proposed simply dividing the scale below reference into equally sized slices. As I said, that was not the recommended method early on. It was sort of treated as a last resort, but somewhat alarmingly, about half of all Water Framework Directive methods seem to be using this now. And then as implementation progressed, more countries were setting boundaries, and with intercalibration, which I'll talk about in a moment, we had some sort of confidence, there was sort of agreement amongst the countries. The alternative possibility arose that we could actually use this consensus of approaches as boundaries for those countries that were joining the exercises late. And that could also be seen perhaps as a sort of a shift of emphasis away from the sort of elusive hunt for the holy grail, which was the unimpacted condition, towards a more pragmatic focus on ensuring consistent application of the key regulatory benchmark. So here, just as an example, is how we're setting boundaries for the original UK phytobenthos method. We set our high good boundary as the 25th percentile of reference samples. Our reference screening at the time wasn't as elaborate as the example I showed on the earlier slide, so we felt that the reference subset weren't all truly referenced, so a 25th percentile would be a feasible high good status boundary. We used the crossover approach to set the key regulatory boundary, which is good moderate, taking the TDI apart to yield nutrient sensitive taxa, nutrient tolerant taxa. And then the scale below that, the 2 divisions below that, less important from regulatory purposes, we set by dividing the remaining scale. And then I'll come on to this topic of intercalibration, because it's something that's happened in Europe, and it's clear from our discussions, nothing like it has happened, at least not on the same scale in the US. It was managed by Ecostat, and it's performed separately for each biological quality element. And it happened in 2 stages. The first was a compliance check. We looked at the national methods developed by the member states to ensure that they were following the wording of the Water Framework Directive, and especially that they were following the narrative descriptions of ecological status as they were setting up their methods. And then this was followed by a comparison of boundaries with those of neighbouring countries. It only happened for the upper two boundaries, high good and good moderate. And to do this, countries were grouped into geographical intercalibration groups, or GIGs. So the theory was we were comparing like with like. And then some of the larger countries were in 2 or even 3 GIGs. So France, for example, was in an intercalibration with UK, Germany, and some other sort of Central and Northwest European countries. But it was also in the Alpine GIG, where they were looking at the sort of mountain streams, and also in the Mediterranean GIG. So you can see by the time lots of countries have been placed in 2 or 3 GIGs, you end up with a sort of a web stretching across Europe where everybody's sort of checking themselves against 2 or 3 other groups. And the way this happened was that we, first of all, we had to express all of our national metrics on a common scale. So we set up some sort of simple intercalibration common metrics for the different quality elements. And for phytobenthos, this was the average of the EQRs based on the IPS and the TI, the 2 most widely used metrics. One of them sort of one of them focused on the whole gradient, picking up saprobic effects. One of them focused just on sort of presumably or inorganic nutrient effects. So then you could take the boundary value of, boundary value of each national metric, and then by a simple process of regression, convert that to the equivalent value of the intercalibration metric. And then you have a value that you compare with all the other countries in your geographical intercalibration group. So the grey band on these 2 graphs, these are for high good and good moderate respectively. The grey band represents the mean value plus or minus a quarter of a class, a quarter of a status class. And this was referred to as the acceptable zone. It doesn't have a sort of strict quantitative basis, but it's a consensus of what experts from a reasonable number of sort of different biological organism groups thought was a reasonable deviation. Member states, as far as the European Commission is concerned, are entitled to set themselves stringent standards. So we found Austria, we found in this particular case, you can see Belgium actually went into the exercise. Northern Belgium, Flanders, and Southern Belgium, Wallonia, went into it separately. Both of these had quite high boundaries initially. Flanders decided it was going to stick with its high boundary, but Wallonia was a little bit alarmed and sort of wound its boundary down a bit to the top of the acceptable zone. And then at the other end, countries that fell below the expected zone were acceptable zone were expected to raise their boundaries. That was the requirement of the intercalibration process. They could show that their boundaries were consistent with those of other countries. So as you can see in this case, the Netherlands in particular saw the case for raising its boundary. Sort of Sweden made a small adjustment as well. So just to reflect on the intercalibration exercise, it's something that was a massive logistical and technical challenge, but it's a really important given the nature of the EU, because it reconciled the sovereign rights of member states with the collective ambition of the Union. And early on, this was a really great opportunity to share ideas and develop approaches. People coming together, sort of discussing which metrics to use, how to set reference conditions, how to determine status class boundaries, and so on and so forth. And from those discussions, you could say like a supermarket of tried and tested approaches evolved. But that possibly sort of haunted us later on, because later on, it's sort of, I feel, and I know Sandra in our discussions about the paper sort of disagreed with me, it sort of encouraged a sort of conservative approach, particularly as sort of smaller and less well-off countries in Eastern Europe joined the exercise. I think the feeling was, you know, why why spend a lot of time developing innovative new approaches when we can just sort of pick the IPS off the shelf and divide our boundary into 5 equally sized sections and call that our national method. So I feel that in a way intercalibration sort of encourages conservatism to some extent. It's a particularly helpful exercise where countries shared catchments, because you are each country has a responsibility for regulating part of the catchment, and intercalibration should ensure that they are all sort of managing it to the same standards. And it's an ongoing process. So Croatia joined the EU in 2013, so it's going through intercalibration now. Some countries were slow to develop approaches for some BQEs, and so they're joining late. And some countries are revising and updating their methods. And there's details on the European Commission's website on the statistical protocols you have to go through in order to do this. And having sort of mentioned innovation, sort of brings me on to the next section of the talk, which is metabarcoding, which has been it's a big topic everywhere, but it's been a hot potato in Europe over the past few years. And what I wanted to do in this talk was just focus on a few lessons that we've learned in Europe about the application of metabarcoding to ecological assessment, and in particular to diatoms. So the big challenge when you're starting to think about metabarcoding is getting a library of reference barcodes. And lots of individual efforts in Europe are now coalescing around this large database that's coordinated by Frédéric Rimet and his colleagues in France. It's got over 1,000 species represented, which is great, and it's really well curated. That's the other side of it. These are you could just use GenBank if you want, but there's an awful lot of rubbish there, whereas Diet Barcode is a well curated database. But with 1,000 species, it's still only a small part of Europe's diatom diversity. We've got 2,800 recorded species in the UK, for example. So a lot of the discussion has been on how to sort of expand this database. And I think for some other organism groups, sort of the invertebrates, macrophytes, vertebrates, there is a sort of a genuine sort of feeling that they could potentially complete their reference barcode libraries. But for the microscopic organisms, I think we're beginning to realize that's probably unlikely. The diet barcode is biased towards taxa that grow well in culture. Those that grow easily under lab conditions, we could say is biased towards weeds, perhaps. And I would say, having used the term weeds, the problem is one of diatom horticulture rather than necessarily taxonomy or genomics. Sort of how do you get the sort of the more slow growing things to sort of come on in culture? I should say there's examples such as like single cell PCR out there, but one of the advantages of cultures is that you yield material for traditional taxonomy, so you can sort of move forward without sort of making a complete break from Linnaean binomials. So there's two questions. Are there any novel ways that we can expand the data sets or the reference libraries that don't rely on culturing? And the second question is, well, how important is it anyway to complete the barcode libraries? So the first question, alternative approaches, this is one that we've tried both in the UK and in France, is can we identify barcodes directly from metabarcoding outputs? And the approach here would be that you identify a target taxon in a light microscopy sample, find something in your light microscopy that's not represented in the barcode library, and then try and match that to corresponding read in the metabarcoding output. So here's an example. Frédéric had a light microscopy sample. He found Halamphora garnensis, not something in the barcode library, went to the metabarcoding output, used sort of phylogenetic methods to identify the reeds that corresponded to Halamphora, and from that was able to identify a sequence that was equivalent to Halamphora garnensis. Now, to do this, you need matched light microscopy and metabarcoding samples. That hasn't been a problem because in the early stages of development of methods, we've tended to sort of try and equate light microscopy and metabarcoding. You need good sequence quality, you need a high abundance of the target taxon in the light microscope output, and also importantly, you need a sample where there's no close relatives that are going to confound interpretation. It'd be much more difficult if there were sort of two Halamphora species, for example. One of the drawbacks we got here is that the current metabarcoding platforms such as MySeq don't yield full-length sequences. Sorry, I should have mentioned we're mostly using the RBCL gene in Europe. This codes for rubisco. It's a key photosynthetic enzyme, and it's the most widely used marker gene. It's 1,400 base pairs in length. The MySeq only uses sequences of around about 400 base pairs. So the barcodes we identify using this technique, they're fine for ecological assessment, but they're not so good if you wanted to do fundamental taxonomy. But then the other question I asked was, do these gaps in the reference library matter for ecological assessment? I'm answering this by, I guess, what you could call a thought experiment more than an experimental demonstration. It's in a paper that came out this year, which I'll talk more about in a moment. So this paper, to answer the question about do gaps in reference libraries matter, I'm not using metabarcoding data at all. I'm using light microscopy data. I'm using about 1,500 samples here. And on the x-axis, we've got the latest iteration of the TDI, TDI5, calculated with all the taxa that we identified using light microscopy. On the y-axis, again, we've got light microscopy data. Again, we've got the latest version of the TDI, but this time just using the taxa that are represented in our barcode library. And you can see pretty good agreement most of the time. You've got a few outliers, but on the whole, the difference between using all taxa and just those in our reference library is pretty small. So on that basis, I would say the gaps are probably less important than some of the other issues that I'm going to talk about on the next few slides. Now, you might be aware of Donald Baird's proposal for something called Biomonitoring 2.0, taking full advantage of metabarcoding capabilities to develop a new generation of metrics. We had some sort of discussions in Europe, again, across all organism groups and across different water body types, about the pros and cons of Biomonitoring 2.0. We came up with the general consensus that maybe we should start with a less ambitious objective, and in particular try and understand how metabarcoding outputs relate to the data with which we are already familiar. So how, for example, does the representation of individual species vary between light microscopy and metabarcoding? So this is the UK study that I mentioned a couple of slides ago. And here, again, with matched data set over 1,000 samples, is the relative abundance of some common taxa in light microscopy on the x-axis and metabarcoding output on the y-axis. So small single chloroplast species such as Acanthidium, Minutissimum, and Amphora pediculus tend to be much better represented in metabarcoding, in light microscopy output, form a higher proportion of the total than is the case in metabarcode. By contrast, larger species, often with multiple chloroplasts, Navicular lanceolata, Melocyra varians, Ulnaria ulna, the opposite's true, tend to find a higher proportion of the RBCL reads than the number of valves recorded in light microscopy. And then sort of bucking that trend, we've got Fistuliferous saprophylla, which again tends to be better represented in metabarcoding than in light microscopy. Here it's a small species, but it's very weakly silicified, and we suspect it's often not surviving the preparation process, so we're probably under-recording it in light microscopy. Now, at the same time we were doing this, the French team, Frédéric Rimet and co, were looking at the same issue, but using laboratory cultures. And they found a relationship between the copy number of the RBCL gene and the biovolume of the cells of the different species, the Acanthidium, Minutissimum, Nitsia, and Conspicua, representing the smaller cells, Ulnaria, ulna, Pinularia, viridiformis, representing the larger ones. And the first point to make, I think it's an important finding from the last few years, it is possible to produce quantitative data using metabarcoding. And I think, there was a stage a few years ago when people were saying, well, maybe we've got to develop a new generation of of models based on presence absence, because we're not sure if you can quantify. I think the message coming out of Europe now is that you can quantify, and we can think about sort of quantitative models. But the second conclusion from this, I'm going to throw this down as a challenge to the Diatom Web Academy, and you can shout at me in the discussion session later on, is could it possibly be that metabarcoding yields more ecologically meaningful outputs than traditional light microscopy? Because we are quantifying a key photosynthetic enzyme here, rather than simply counting valves. So could that not be giving us a better insight into the contribution that each taxon is making to primary productivity than we are getting with our current approaches based simply on numbers of valves of individual species? However, one implication in the short term is that we don't get particularly strong matches between current and metabarcoding metrics. So this is the UK study I was referring to before. That's our latest iteration of the TDI based on light microscopy. And this is a new version of the TDI that's been calibrated directly against the metabarcoding output. Now, that's as far as most ecologists are concerned, that's quite a nice looking graph. But when you hand it over to a regulator, they get a little bit alarmed because a lot of sites are changing class as a result of the change of method. So we found about 63% of samples changed class, which is, well, sorry, 63% of samples did not change class, but that in turn means about 37% did change class. And this is for samples. When we did it for sites, the situation's slightly better. It's about 70% have got an exact match and 30% a shift in class. But this is still a big problem for regulators, because the programs of measures that I mentioned earlier might well be based on existing classifications, and nobody is going to be particularly keen on the idea of sort of changing sort of investment programs that might run into millions of pounds simply because we've changed the method. So at the final hurdle, the UK government was very reluctant to change classifications simply because there was a shift in the method. And sort of as a coda to that graph, we've also subsequently realized through some sort of collaborative studies with European colleagues that the metabarcoding samples might not survive for quite as long as we thought in storage, and that we maybe, if I prune this data set down to remove samples that have been stored for more than 3 months, we might have trimmed away some of these outliers. I haven't had a chance to do that. And another coda, I suspect the UK government would have been more enthusiastic if the metabarcoding had led to a significant reduction in costs, which had been predicted at the outcome of our project, but didn't actually happen when push came to shove. So ironically, uptake of metabarcoding is a lot easier if there isn't a pre-existing method against which it can be compared, or if it's part of a bigger shift in ecological regulation. So I think some of the other European countries, Germany and France, are thinking that maybe the shift to metabarcoding should come at the end of the next cycle of Water Framework Directive, when there will be a much bigger rethink of approaches. Sort of generally, the whole regulatory process will probably be re-evaluated. So in the chaos that ensues, no one's going to be that bothered if there's some sort of methodological changes. And one final incidence, one final metabarcoding study from Europe that I wanted to share with you would be this comparison amongst bioinformatics pipelines used in 6 European countries. Now, I guess the bioinformatics pipeline is a sort of the computer programs that process the output from the barcoding platforms like the MySeq and turn it into sort of lists of species. So roughly it's equivalent to the brain of the human analyst. So for that reason, I'm comparing the comparison of bioinformatics pipelines, different bioinformatics pipelines of these ring tests where sort of human analysts have sort of compared their outputs. I think Maria Callett's spoken to the Web Academy about this, the Scandinavian ring tests. So they had 25 or 20 or 30 different slides where analysts have compared their results. In the UK, we've done something similar, 50 or 60 results. And these are the typical standard deviations we are getting from sort of human analysts or experienced human analysts comparing their samples. And I was quite surprised to see how different or how much higher the standard deviation was when you applied exactly the same approach to this comparison of bioinformatics pipelines. And this was done for both RBCL and for another marker gene, 18S. Emphasize here, identical input data, identical reference library, but quite a lot of variation between bioinformatics pipelines. I don't want to dwell on these graphs too much. This is an example of how Fragilaria varied both between 18S and RBCL and between different pipelines within the using the different markers. So you can see both a variation in the proportion of Fragilaria and variation in the species that were identified. This is again, bear in mind, the same reference library used for all of them. There was actually a sort of a hitch, we realized, which is why we got no Fragilaria at all in the French pipeline. That's mentioned in the paper and more as a salutary lesson than as a serious result, and similar results there for Eunotia. So this pipeline comparison, identical input data, identical reference libraries, but significant differences in outputs. Now the short-term option is you include the specification for pipelining your standard operating procedures. That's sort of what our the officials and the regulatory organisations in the UK thought would be the best way to proceed. But longer term, this raises quite a lot of concerns. You make some decisions when you set up a bioinformatics pipelines, but at this stage, I'm not sure that we really understand why there are differences between pipelines and which of the many options in the pipeline are giving us the most honest interpretation of reality. Bear in mind at this stage that light microscopy has its own inherent biases, so we can't simply look for the pipeline that gives us the closest match to the light microscopy output. So this is where we are with metabarcoding in Europe. That we started out this option 1, trying to replicate current assessment approaches. The French group have been advocating the use of correction factors based on the study between biovolumes and RBCL read numbers that would allow us to align metabarcoding results with existing metrics. And in the UK, we've tried to develop new metrics directly from metabarcoding data, but using existing assessment concepts. I think this has been a very useful procedure. And I would say we are beginning to understand how metabarcoding data relates to the current knowledge base. So maybe now we are in a position where we can start thinking more seriously about biomonitoring 2.0 and sort of opening up the full potential of metabarcoding as we go forward. So to summarize what we've learned from metabarcoding studies in Europe, well, we started out with the metabarcoding evangelists telling us that it was going to be faster, cheaper, and more precise. and haven't got time to go into all of this, but I would put a question mark behind each of those claims on the basis of 7 years experience. There's a fundamentally different mode of quantification, which means you're always going to get sort of differences between light microscopy and metabarcoding. I've talked a little bit about the gaps in reference libraries, some ways we can plug the gaps, but also I think we need to be a bit more realistic about whether that's feasible and whether we really need a complete reference library in order to move ahead with the type of ecological assessment that we're doing. I think also this is an example, really good example of the benefits of collaboration, because I think we've achieved far more as a group than any of us could have achieved individually. And one of the fruits of that collaboration, we're working now with the European Standards Organization to make statements about best practice that can serve as a platform for moving forward. I haven't sort of mentioned this, I sort of just toss this in as a slightly provocative comment, but we started out looking at diatoms almost as a sort of as a test case, a proof of concept. But at this stage in the process, I'd say, why are we limiting ourselves to diatoms? We limit ourselves to diatoms for the light microscopy because there's particular issues about the way you prepare diatoms and about their identification that means it's difficult to study diatoms alongside other groups. Those limitations don't apply when you're using metabarcoding, so why not start out with a broader scope? And finally, when you're working with regulators, I would say one of the things I didn't anticipate was that this is really a case of organizational change that has to, and the importance of the changes that need to be made across an organization need to be considered. At one level, you are switching from a sort of an expectation of an ecologist being someone who goes out in the field wearing waders and with a pond net to a situation where ecology is done by people in very clean laboratories wearing lab coats and surrounded by very high-tech equipment. And also the second aspect, so just the whole nature of ecology and ecologists, and sort of almost the sixth sense that an ecologist develops through being out in the field and working through samples. But where's that going to come from in the metabarcoding era? But also we've seen huge changes in metabarcoding in the time that we've been involved with it. And our regulators are very keen that they have a standard method that they can use for years into the future. Whereas I think in the metabarcoding era, we need to find ways of being able to incorporate new developments into practice that don't compromise regulatory needs such as consistency and transparency. And in a way, I think there's a lot of the methods we need are available via the intercalibration process. In intercalibration, we were looking at harmonizing across space. In this kind of, in metabarcoding, I think we're going to need to harmonize across time, simply to make sure that the regulatory outcomes are the same, even if the underlying processes have improved. And then bring me on briefly to my very last point. I think metabarcoding is a good platform to move into the communication bit, because it's taken us, taken the scientist one step further away from a capacity for easy interaction with the public, much more abstract type of data. Now, I think this has always been a problem when you're dealing with periphyton and diatoms. Metabarcoding simply accentuating this, convincing, I mean, the lay public, stakeholders and managers are concerned with sort of fairly gross effects, such as filamentous algal growth and what that does for water users and for perceptions. How do we convince regulators and lawmakers that assessment based on a group of organisms that they know little about are actually worthwhile? And I don't think diatomists always help their cause with their excessive attention to detail. And I think the macrophyte scientists in Europe are setting us a really good example. So they've done all the hard quantitative work that I've talked about on the earlier slides, in terms of reference conditions, creating metrics, setting boundaries, intercalibration, so on and so forth. But they've also finished off by taking the narrative descriptions of ecological status and using these to create guiding images that then make it easier for stakeholders to understand and appreciate what they're paying for. So just to sort of finish off with my sort of concluding statement, if you asked me 25 years ago if I would be telling you half of what I've been telling you now, I'd have probably laughed at you. The whole idea that periphyton could be part of a formal regulatory system with an underpinning legal framework and responsible for decisions that cost millions of dollars, euros or pounds, yeah, that would have been unbelievable. The early meetings we went to on the use of algae for monitoring rivers that Eugen Roth and Brian Witten organised, largely talking about the possibilities and potential. It seems like 25 years on, you know, we seem to be getting there. But that in turn, it's creating responsibilities that go beyond simply creating strong models. In a small way, our own small way, we're part of the democratic system now. We've got responsibilities to regulators who are responsible in turn to elected officials. And those responsibilities include transparency and communication, because we've got to sort of explain to people why their utility bills are increasing. So in a way, we're sort of playing with the big boys now. I didn't expect that 25 years ago. But I think it's going to, we need to move the conversation on, sort of grapple with some issues that we have perhaps felt have been somewhat outside our comfort zone until now. So that's me finished. So thank you for listening. We look forward to your questions and comments.
Thank you, Martyn, for a great talk. And I see that there are a few questions stacked up in the chat. So I'll go ahead and start with some of those. First two are related. First from Susan Jackson. For those countries that were asked to raise their boundaries, such as the Netherlands, was attainability of higher conditions considered in any way?
No. This was there are sort of checks and balances in the Water Framework Directive that I haven't really got time to go into now. It is possible for a country to set less stringent objectives if set good ecological status is deemed to be technically infeasible or disproportionately expensive. So the Netherlands in that case would have had to set a realistic good status boundary, but then make a case for only achieving moderate status. So good status should be a sort of like a pure concept. And then there's other checks and balances within the directive that would let countries such as the Netherlands get round that particular situation. And I have to, well, I should admit that the United Kingdom was the most blatant player of that particular get out of jail free card. But then we've just got out of the whole European Union, so much to my regret. Don't want to get onto that, do we?
So, and Sylvia has a kind of a follow-up question, maybe you've answered it, but how agreeable were countries to raising the good moderate boundaries, and was there pushback from stakeholder groups?
I wasn't party to individual decisions such as the Netherlands, so I can't answer that. I suspect there would have been. Certainly every time we propose a new phosphorus threshold in the United Kingdom, you know, the stakeholder groups are definitely sort of pushing for more lenient thresholds. So I'm sure there was, yes.
And from Jan Stevenson, great talk, Martyn. It's impressive to see the pioneering work by Europeans on so many of the algal assessment questions, and has been history over the last century.
Thanks, Jan. A lot of the time, I don't know if you notice, a lot of the European studies are based on sort of association statistics, not experimental studies. And I think we rely upon the foundation of experimental periphyton studies on your side of the Atlantic. We're very grateful for that.
And from David Burge, as bioinformatics are evolving, is there a data curation plan? And then have you explored combining reference databases and phylogenetics to fill taxonomic gaps?
Well, cards on the table. I'm not a bioinformatician or really a molecular biologist at all. Data curation plan, again, I can only talk for the UK. There are sort of plans to sort of, to store the raw metabarcoding output so that we can revisit it as understanding of bioinformatics and what have you evolves. Combining reference databases and phylogenetics to fill in taxonomic gaps. Can I duck that question because it's a bit outside my comfort zone?
Well, can I ask a related one then? And how about actual samples in the deep freeze? Are those archived for genomics?
I think in theory, yes, but I think well, first of all, the actual samples, we have found some deterioration in samples in storage. So I think in theory, samples are being stored, but as time goes on and fridges fill up, and particularly as organization sort of restructure, I don't think you can ever guarantee, unless it's in a formal herbarium whose mission is the preservation of biological materials. I never quite trust sort of government regulators to be as such good custodians as you would expect a sort of a herbarium to be. So I'm not sure if that's probably a partial answer. It won't be me that throws them away, is what I'm trying to say. But it's not my fridge that's full up either.
Jan asks if you could elaborate on the concern about rapid climate change and the reflection into algal and diatom metrics. Is there evidence, either empirical or conceptual, or for this rapid change?
There is some. I examined a PhD a few months ago, just at the start of lockdown, and that showed some quite significant changes in the diatom flora over 25 years or so, based on some samples that Roger Flower had collected and stored, and showed quite a lot of changes. And one of the biggest predictors of change was hydrology. Unfortunately, that PhD is still in the sort of rewriting phase as far as I know, so I can't say much more. The case in I mentioned in Romania was that just looking at about 5 years of conductivity and alkalinity data, there was a sort of a definite upward trend in many of their shallow lakes in conductivity. I didn't really have a chance to do a detailed analysis to see to what extent the biota was changing, but there's enough of a relationship between conductivity and diatoms that I'm sure there would have been. I think it's an area that we need to do more work on, and it's actually an area where the Water Framework Directive is pretty weak, because the Water Framework Directive is 20 years old, so it rarely mentions climate change directly.
Okay, there's a couple of more bioinformatics questions. I'm going to skip those and come back. I want to get to This question, follow-up question from Susan. Do you foresee the results or scores for diatoms used independently in making a decision on the condition of a water body, or weighed with the results of benthic or fish assessments? Or, you know, maybe more realistic, has there been this discussion among ecologists you work with on the application of these multi-assemblage assessments?
Well, I'll answer it two ways. First of all, in old-fashioned Water Framework Directive terms, sort of the one out, all out rule means that diatoms are always part of a suite that contribute to a decision. But what I find, and I find that if the periphyton is the only factor that fails, the regulators are quite cautious about taking an expensive decision about sort of upgrading wastewater treatment works simply based on a diatom failure. In theory, they should, but I think they feel that even if they have to classify a water body just on the basis of the diatoms, then they would probably resort to a stock phrase like, or the first step of a program of measures often turns out to be collecting more data, rather. So they sort of they try to kick the can down the road. I think they are reluctant because members of the public can walk past a water body that's at moderate, in my opinion, they can walk past a water body at moderate status and not know there's anything wrong with it, because we set quite a high bar. And particularly if the substrate doesn't let filamentous algae grow, if you've got like a fine substrate, you know, the public wonder why on earth their water bills are going up. And I think the regulators are reluctant to make decisions even when the diatoms are pointing in that direction if there's not another indicator. That's my personal view. The question about multi-assemblage assessments, there is a paper came out, I was just a sort of a minor contributor to it, sort of almost suggesting that we could start combining sort of metabarcoding output for invertebrates and the metabarcoding output for diatoms, and sort of trying to sort of think in terms of food webs. You think in terms of making a status assessment based on the ecosystem rather than sort of separate assessments based on each organism group. So but I think that will come, because if you think of the work on the different organism groups and the work on the bioinformatics and the other people that are working on food webs, you could see a sort of a new paradigm emerging. If we did that, we'd have to move to a different marker gene that was a more sort of universal marker gene. So I think you can probably tell I'm waffling now, but
well, our time has run out, but the questions keep coming. Martyn, if you're okay, if people
I'm happy
email you with their questions, is that, urge
well, I'm fine, yes, I'm not a problem.
Get your question answered, please contact Martyn. And I don't know if somebody, Mark or Sylvia, can pop your email in the chat right now.
Can I just answer Tom Frankovich's question about the cost of producing DNA library for?
Oh, sure.
Because this is a funny one, I think. Well, the answer to that is a lot. I mean, I think we had one postdoc for a year, and he did 100 species, and he worked his socks off. But one of the things we got out of the big data set was with Maria Callett did some taxonomic work on Fragillaria, and we found 3 new species, and 3 of us named those species after our partners. So one of them, I gave it to my wife as a birthday present, and I said it was both the cheapest birthday present I've ever given her, because effectively all I did was put a picture of of the diatom in a frame, and the most expensive, because there's probably at least £500,000 of research behind that database behind it. So that's a sort of answer to Tom's, but I think, yeah, these databases are going to be really expensive to complete.
Okay, well, thank you everyone for joining us and for the great discussions and questions. And thank you, Martyn, for being willing to present. We really appreciate these connections in these dark times and being able to join colleagues to get together. So Becky Bixby will be speaking in 2 weeks, which is Tuesday, January 19th. Same time, same channel. Becky will be talking on the role of light availability in shaping diatom assemblages. So stay healthy, be safe, reach out to colleagues, and this is a way that we can continue to do interesting work and connect with each other and support each other and find the silver linings in these dark times. So thank you, Martyn, and thanks everybody, and see you next time.
Thank you.